Je m'appelle Mariam et je suis la troisième année de ma thèse. Je vais présenter en anglais parce que c'est plus facile pour moi et je pense que je suis beaucoup plus claire qu'en français. My thesis is mostly about dose finding in phase 1 and in oncology. But today I will talk mostly about what is the importance of taking into account in dose finding the biomarker trajectory that we use to measure the activity of the drug. And in this case, we use joint modeling. We have a lot of missing data. And as you said before, in this case, our sensitivity analysis is mostly a problem rather than a sensitivity analysis. So before going to the joint modeling, I will describe the kind of data that we have. So in a phase 1, we have the patients that enter the study at time 0, and they are followed for a treatment cycle that could be between 3 and 4 weeks. At the end of the treatment cycle, which is denoted with C1, patients will come and we are interested in the kind of toxicity events that they had, and mostly the grade 3-4 that could lead to dose-limiting toxicity, and as a result they can get out of the study. However, during a treatment cycle, patients will be measured multiple times regarding the activity of the drug. Now, one way to measure the activity of the drug is by a certain biomarker that it is related to the disease, and we do that with blood sampling. Again, at the end of a treatment cycle, if we see that the drug is not active anymore for the patients will get out of the study. So once again, we have missing data. Of course, this is not so much the case in the first treatment cycle, but in the next cycles. Of course, patients can also get out of study because of reasons of consent withdrawal, which, however, are more rare in phase 1 trials. If none of these things happen, then the patients will go to the next treatment cycle and we will repeat the same procedure. So in our design, we wanted to address two major issues that we have in dose finding. Now, the first one is that when we want to find the maximum tolerated dose, we only take into account the toxicity data that we have, and only for the first treatment cycle. Now, the problem here is that we completely ignore a late onset toxicities that we might have, and toxicities that accumulate when we have a long follow-up. A problem in phase 1 trials is that we have seen that by the end of the 6 treatment cycles, 93% of the patients will get out of study. However, this is not the case in phase 2, and we work in molecularly targeted agents which are administered for a really long period of time that can go above 20 treatment cycles. So we have cumulative toxicities. Another problem when we work in phase 1 is that we completely ignore all the activity measurements that we have when we want to find the maximum tolerated dose. Now, here I show you the DLT target database that was created by the EORTC, and it consists from I think 54 trials that come from academia and from pharmaceutical companies. And in my case, I analyzed 27 of them that come from the NCI. And these phase 1 trials are completed, and they have molecularly targeted agents as monotherapy. Now, for these trials, we have 963 patients, and we have all the information of their follow-up, the cycle of treatment, the dose that they were administered to, all the toxicities that they had, and the grade of the toxicity. Now, the first thing that we wanted to do was to evaluate what is the conditional and the cumulative probability of having a toxicity in the first 6 treatment cycles, and more specifically in the results that I will show you here, it's for the MTD. So in the first table, you can see that we have the conditional probability of having a grade 3-4 toxicity per treatment cycle. The reason that we have the grade 3/4 toxicity and not the DLT is because the DLTs were only registered for the first treatment cycle and not for the rest. So as a result, we were obliged to take the first grade 3/4 toxicity. In the second table, you can see what is the cumulative probability of having a grade 3/4 toxicity at the MTD. Now, normally, when they take the first treatment cycle, they say that they want to have between 20 and 33% of adverse events. However, if we go all the way to the sixth treatment cycle, we can see that we have 53% of cumulative toxicity, which is a lot, and we need to take that into account at least for the subsequent phases. That is the first issue that we have in dose finding. Now, the second one is related to the biomarker trajectory. Now, typically, when we work with cytotoxic agents like chemotherapy, we are interested in the maximum tolerated dose. And here you can see a figure where we have the dose here and the probability of activity in, with the dashed line and the probability of toxicity with the solid line. Now, these two figures have pretty much the same shape, and as a result, we know that the higher the dose, the better activity we will have. However, this is not the case with targeted agents. Here we can see the trajectory of the biomarker. Again, we have the dose and the probability of activity. Now, the difference here is that after a certain dose, the activity reaches a plateau. So that means that even if we increase the dose, we will not gain something in terms of activity. So the question we wanted to address is that if we know that the MTD is here, where we have the vertical line, is it possible to find a dose that we call an optimal dose that is as active as the MTD, but it is yet less toxic, which is very important when we work with cancer patients. And as you can see here, we have certain doses that are on the plateau and have pretty much the same activity. So in reality, if we have a dose that we think that it is the optimal dose, it would be where the plateau begins and not where the MTD is. So that was the second issue that we wanted to address. To do that, we proposed an adaptive design. I will not talk so much about this design today. And the objective was to define the maximum tolerated dose, but cumulatively over all treatment cycles, and then to identify the optimal dose that would be the lowest dose within a range of highly active doses. And this dose can be the MTD or it can be below the MTD, but we do not allow to exceed the MTD. Since we are interested in both toxicity and activity, we need to combine data of time to first dose limiting toxicity and biomarker activity over several treatment cycles. The best way to do that for us was joint modeling, and specifically joint modeling of interval sensor time to first DLT data and repeated and continuous biomarker measurements. Now, in this joint modeling, these two models share the random effects. Before continuing with the model, we have been asked many times why we do the joint modeling. You mentioned before how much can it add to our actual estimations in terms of missing data. Now, one of the main reasons that the joint modeling was created was for the survival outcomes. In many cases, we have what we call endogenous time-dependent covariates. And one of the ways, if not the only way, I do not know that, is to model this endogenous time-dependent covariates is to do joint modeling. So in practice here, when we have the clinical trial, even though we might initially be interested in the toxicity, we still lose patients because of disease progression. And disease progression is something that is sure in phase 1 trials. So once patients progress and they get out of study, we no longer have information on their biomarker profiles. So this is what we call an endogenous time-dependent covariate. And as a result, Now, the second reason that we want to do the joint modeling is the other way around. Because we have a lot of DLTs in phase 1 trials, after some point we do not have any more information for the biomarker profile of the patient. And as a result, we have missing data. Now, we can use the joint modeling to take into account for all the missing information that we have, and also we have a lot of information, toxicity, activity, multiple treatment cycles, so we think that it is better if we take that into account before going to phase 2 trials. Now, one of the most famous joint modeling techniques is by Rizopoulos, that was mentioned also before. However, this is not the models that we used. So we'll start from the activity. So here we can see the model for the activity endpoint. We have continuous and repeated biomarker measurements, so we took a linear mixed effects model. In this model, we have adjusted for the time effect, which is the time that the patient comes to measure the biomarker, and also for the dose effect to which a patient is allocated to. We have taken the log of the dose in order to make the biomarker curve to be a little bit smoother. And the time in this case is continuous. Now, one question that we were asked a lot is why do we take the time to be squared and not a linear term? So here we can see a figure of a biomarker, CA125, that it is used a lot in ovarian cancer. Now, this biomarker decreases when when the drug is active, so patients are going well. And after some point, the biomarker starts to re-increase because we have disease progression. In this figure, you can see the CA125 levels with the percent of progression-free survival. It comes from a trial from Zhu and colleagues, and it shows the dynamics of this biomarker. So this is the reason that we model time with a square. We wanted to capture this effect in time. Because if we take a linear term, then we will completely ignore the disease progression, so we will only have the time where patients actually respond to the medication. The next model for the joint modeling is for the survival outcome. In this case, we have a probit model here. And we have adjusted this model once again for the time, which in this case is the cycle, and also for the dose to which a patient is allocated to. Here we can see the random effects that come from the linear mixed effects model, and these two models share the random effects via a gamma parameter here. For the toxicity outcome, we have an interval censored model, which is nothing else but the treatment cycles. Now, one of the major issues when we do the joint modeling is related to the parameter estimation. And this is because normally we have integrals and we need to estimate these integrals, and if we have one random effect, then it is easier, but when we want to take into account more than one random effect, then we have a high-dimensional variance-covariance matrix, and as a result it gets more and more difficult to estimate the effects. Now, in our case, we took a relatively new joint modeling technique that was proposed by Barrett and colleagues, and they took advantage of certain properties of the skew normal distribution in order to integrate out the random effects and write the likelihood in a closed form. So in that case, in order to get our parameter estimates, we only need to do a numerical maximization, and we don't need to approximate anymore. As a result, we have really good parameter estimates. The authors have shown a very good bias and coverage. However, they showed that in a sample size of 1,000 patients. So the first thing that we did was to test how well this joint modeling technique works when we have really small sample sizes, as is the case in phase 1 trials. And we've seen also that it has really good bias and coverage. And we can start to use this joint modeling technique once we have 20 patients in our data. So we use this joint modeling technique every time we have a new patient in our data. So before allocating a patient to a certain dose level, we run the whole analysis and we find what is the better dose. To do that, the first step is to start from finding the maximum tolerated dose. So here we have a figure where we have 4 dose levels and the cumulative probability of toxicity for each dose level at the end of the trial. So here, for instance, dose level 4, we know that at the end of the trial will give us 80% of toxicity. Now, before even starting the trial, we need to define a threshold that we either do not want to exceed or we want to be as close as possible. So in this case, our target is 40%, and the maximum tolerated dose will be the dose that will give a probability of toxicity that is closest to this threshold. Now in this case, dose level 3 is the one that is closest to our target. So we declare dose level 3 to be our maximum tolerated dose. The next step is to find what is the optimal dose. And as I said before, we cannot exceed the maximum tolerated dose. So we will be, this dose will be dose level 3, 2 or 1. To do that, we need to look at the biomarker trajectory. Now here we have the 4 dose levels from before and the relationship between time and the biomarker measurements. So as I shown you before, in the beginning of the trial, the biomarker decreases and then at some point it starts to increase back. So what we want to do is for each dose level, irrespectively of what time we see that, to find what it is the minimum biomarker measurement, because the minimum biomarker measurement is related with the maximum activity. So for the 3 dose levels that is for interest to us, we measure at each time what it is the biomarker measurement. So 1 dose or more can be equally active as the MTD if they fall within a predefined range. So here we have a range of doses, and if 2 or more doses fall within this range, we assume that they are equally active. So as you can see here, we have dose level 3, which is the MTD, and dose level 2, or the minimum point of dose level 2, falls in this range. So we can say that these 2 doses are equally active. So the optimal dose here that we will recommend for the next patient, or if we are at the end of the trial for phase 2, would be dose level 2, which is also less toxic, as you can see here. So this is how we take into account the biomarker trajectory to make our decisions for the dose finding procedure. And for our analysis, we used, as a motivating example, a Phase 1b multicenter trial in France in patients with platinum-resistant epithelial ovarian carcinoma. Now, this trial has either just started or is about to start, and the primary endpoint is to determine the maximum tolerated dose, and next, to find what is the optimal dose or the recommended phase 2 dose. And to do that, they intend to take into account the activity measurements. For that, for the activity endpoint, in order to define the response and the disease progression, they measure both the tumor volume and this biomarker, the CA125. So we did a similar study where we simulated data that resembled this study. So we had a total of 60 patients, 6 dose levels, 6 treatment cycles, a maximum of 18 visits per patient. In that case, in each treatment cycle, we had 3 measurements for the biomarker. But I say a maximum because it is possible to have intermittent missing responses. So a patient did not come one time to measure the biomarker, even though they are not censored. We allow to have dropouts because of those limiting toxicities, lack of activity or consent withdrawal. The target that we didn't want to exceed was 40% after 6 cycles, and we said that 2 doses will be equally active if they have a maximum difference of 20 units in the biomarker measurements. So here, I'll go fast, it's just some results. Here we have the scenarios, there are 6 different scenarios in this table for the 6 dose levels that we have. And in red, I have the maximum tolerated dose, and in bold values, I have the optimal dose. So for instance, in this scenario, in 80% of the simulations, the algorithm could correctly identify where is the optimal dose. Now, the advantage that we have when we jointly model both toxicity and the biomarker is that these techniques, where we evaluate at each step what it is the best dose, work very well when most of the patients are allocated here on the MTD. However, we can see trials like this one, where the MTD is dose level 4 and the optimal dose is dose level 1. So taking into account the biomarker trajectory allows us to recommend a dose that it is 3 dose levels below the MTD, and yet it is equally active. So this is what we mostly gain by doing the joint modeling and taking both into account. I will not continue anymore on these results because it's not really the objective. I can only talk about how well this the joint modeling works in our case. So what we really gained here is, as I said before, when we have really big differences between the optimal dose and the MTD, it still allows us to correctly identify what is the optimal dose. And of course, we also did a lot of sensitivity analysis where we considered the fact that we might have larger variances than the ones we simulated, smaller sample sizes, but the smallest we could go was 40 patients, because we also had 9 parameters to estimate at each time. We assumed that we might have increasing hazard at each treatment cycle, and apart from generating data from different models, one thing that we were really interested in was to see what will happen if we assume that the 2 models have random effects that come from different distributions. So in this case, we saw that there was really no difference in the optimal dose selection, and I think I had read somewhere in the Rizopoulos book that the random effect misspecification in the distribution does not cause any issue in the joint modeling estimation. So this pretty much summarizes what I said before. What was really important for us in this case was to be able to take into account the fact that we have missing data and the fact that we lose information for one outcome because of the other outcome, but still for something that we are able to observe. And what we think is really important and we would like to do next is to be able to do individual predictions. So with this joint modeling technique, we can take into account the individual trajectory of each patient and be able to do intra-patient dose escalation. This is something that we haven't done yet, and I cannot say if we will see big differences in our data because of this escalation or de-escalation in our data. So thank you very much for your attention.
Merci beaucoup. S'il y a des questions. Alors c'était compliqué, hein. Moi je ne suis pas sûre d'avoir une question très pertinente. Si j'ai bien compris, l'objectif c'est au cours d'un essai thérapeutique de choisir la meilleure dose à administrer. Est-ce que vous avez des données de simulation? Est-ce que vous avez testé ce que ça donnait en conditions réelles, dans un vrai essai?
Est-ce que je réponds en français ou en anglais? Parce qu'il y a quelqu'un qui ne comprend pas français ici.
Comme tu peux.
Okay. We have toxicity data, the ones that I showed you before, that come from the EORTC, but we do not have the activity data that we need in order to see in time what it gives. Another problem that we have is that in most of the cases they dichotomize the biomarkers. So instead of taking the continuous measurements that we have, they dichotomize it so it works, doesn't work, something like that. So that is also another problem that we have. But for now we do not have the activity data that we need. We have the toxicity but not the activity.
Je vais continuer en français moi aussi. Ce que j'ai pas compris, c'est pourquoi le composant de survie ne s'intéresse qu'à la toxicité et pas à la progression dans les premières diapositives. Ce que vous mesurez de façon répétée, c'est le biomarker. Et ce qui est mesuré dans le modèle joint, ce qui est mesuré comme un modèle de survie, c'est uniquement la toxicité. Only toxicity for the survival model. Where is activity or
in the gene modeling?
relapse of the disease?
In what we measure, we have both outcomes. Here, in the linear mixed effects, we have the activity, so the biomarker measurements, and here is the toxicity of the survival outcome.
Pas d'autres questions?
Yeah, I have one question. You say that the joint modeling take account for the missing data or the loss of follow-up, but we didn't know how many patients are following up or are dead, or so do you have a sense of number of these patients?
We try to simulate the toxicity data in the same way as the ones presented from the URTC. So at the end, so that you cannot see it, but at the end of the 6th treatment cycle, around 93% were out of the study either because of disease progression or DLT. And the probability of having an event at each treatment cycle started around 30% and decreased at each treatment cycle. So the censoring was similar. However, after the 4th treatment cycle, a lot of patients, around 50%, got out of study because of disease progression. So there was no activity, so they left the study.
Merci.
Merci.
Pas d'autres questions? On va passer à la dernière présentation.